All right. Welcome everyone to our Lafora Therapy Town Hall at our symposium in 2025 this past month. We mentioned at the end of our discussion about the research into Lafora therapies that we wanted to host a follow-up meeting to give families more of an opportunity to learn about the therapies in development, to ask questions, and to give the Lafora community an opportunity to come together and make some decisions about how we can get more therapies to the clinic for Lafora disease. So to get us started, I'm going to begin with a brief presentation to give an overview of some of the things that we discussed at symposium. So here are our goals for today. As I mentioned, first and foremost, to review what we've learned. And I know that we have a number of members from our research and clinical community joining us today. Thank you for joining us. We really are tailoring this meeting and the message to our families, but particularly as we get into questions and answers, please feel free if you have information that you think is important for our patient community to know about the different therapies. Please feel free to add your expertise when we get to that question and answer time. So we do want to make sure we leave a good amount of time to address any questions that families have about therapies. If you don't see an update for our families on a therapy that you wanted to get an update on, please feel free to put questions in the chat, even while I'm doing this overview presentation. Once I get through all of the updates, then we will make sure that we address everyone's questions. And then our third goal is to have a discussion of how we can accelerate therapy development and get more therapies ready for clinical trials for our community. So to begin, I'm going to do a quick recap of a presentation that I gave on symposium, just to make sure all of our families understand the process that any drug has to go through in order to be approved by either the FDA or the EMA in Europe. So there's 5 general steps to approving a new therapy. There's the initial step, which is focused on discovery, designing the therapy itself. Once the therapy has been designed, you first test it in animal models. We call these the preclinical studies, and that allows researchers to confirm that the therapy does what they think it should do. It also gives them a chance to test what's the appropriate dosage and how long the drug stays effective, so how often they have to administer a dose. All of this gets tested in the animal models. Once the preclinical studies have been completed, the third phase, the third of 5 steps, is the clinical trials. And notice that there are multiple phases. The first phase, phase 1, is to show and demonstrate that the drug is safe in patients. Phases 2 and 3 are focused on whether or not the drug is effective at stopping or slowing the disease. In the 4th stage, that is once you've got the data from your clinical trials, you submit to your regulatory agency, either the FDA or the EMA, for review and if they will approve the drug for all to use. And then there is a 5th phase. Even after approval, data has to be collected and submitted for continued monitoring to make sure that when the drug is administered to everyone within the population, that it continues to be safe and effective. So to help everyone understand what that looks like in terms of how long it takes to get through each phase, this is where it can be a real challenge for our community. The discovery and the preclinical phases can take, on average, 6.5 years. And once you get through these initial tests, the clinical trials themselves, you're looking at another approximate 7 years, and that's assuming everything flows from one phase right into the next. And we know as a community that it doesn't always go directly from one phase to the next. Things come up that you don't expect. So that's why it says on average takes about 7 years to get through the clinical trial phase. And then that final regulatory phase to get the drug approved, that can take about on average 2 years. That's for the FDA. For the EMA, it can take a little bit longer. It can take up to about 3 years. Now, there are some different pathways on the timeline. The regulatory agencies are aware that this is not an ideal timeline for the rare disease community, particularly when it is a rare disease that involves neurodegeneration, involves fatalities. And so there are some accelerated approval pathways that are in development, some that are already in place, that will allow you to run clinical trials and collect data in parallel to speed up that process, to get you more quickly to that regulatory approval process. And so before we get into the updates from each of the therapies that we have as a community within the pipeline, just a quick look, an overview of where some of these different therapies are at. So we have a few therapies that are still kind of in discovery development phase. We have cross-correction amylase gene therapy and enzyme fusion therapy, which we learned at symposium have been developed, been designed, and are now starting to move into that preclinical space. We have a number of therapies that are in the preclinical space, and some have been there for quite some time. And so that includes the enzyme therapy Valo417. It includes a number of different gene therapies. And part of our discussion at the end of this meeting will be which of these therapies do we feel any of them are promising enough that we really want to make a push as a community to try and get them from preclinical into clinical trials for patients. And of course, we do have 2 drugs that have actually been tried in patients. Val-1221 is in red because it was given to about 8 Lafora patients through compassionate use programs. But the data that was collected from those patients showed that the drug did not get to the brain. And so it was not effective at stopping the epilepsy or the progression of the disease. So this drug would have to be modified so that the delivery could reach the brain. And in order to do that, then this would have to move back to some preclinical studies in order to then move it forward into a space where it would be effective as a clinical treatment for Lafora patients. And then the therapy that we'll start with on discussing our updates, the ION 283, which of course we have our safety study that the patient community is currently funding. We'll get some updates on that and what we know about next steps. All right. So at this point, I'm going to pause and switch over to our update on Lafora therapies and what we learned. About those different therapies in the pipeline. So this, again, this is all information for those of you who watched recordings from the symposium. What we're trying to do is just summarize and give you some of the highlights of what we learned about the different therapies and where they're at in development. All right, so again, to give everyone an overview, all of these strategies for treating Lafora disease, we have 2 primary strategies that target the glycogen, target the Lafora bodies themselves in patients. And so the first strategy is we know that these Lafora bodies, as they grow in the cells, particularly in the brain, is what's driving the disease. So if we could send in some sort of molecule, some sort of medicine that acts sort of like a pair of scissors, right, can then come in and just chop up and destroy those Lafora bodies, that's one way to potentially manage the disease. A second strategy is if we can prevent the glycogen from being formed in the first place, or if we can slow how quickly glycogen is being synthesized, then that gives the cells opportunities for different ways to manage the glycogen in the cells, then prevents Lafora bodies from forming in the first place. And so you can think of this strategy, the analogy of water flowing through a sink. So if you've got this flow of water, is all of the sugar that can get stored in these glycogen molecules that turn into Lafora bodies. In a healthy person, right, the glucose comes in, gets stored in glycogen, and then it can be accessed whenever the body needs energy. In Lafora disease or in other glycogen storage diseases, the glucose, right, that flow comes in, gets stored into glycogen, but then it's like you have a clogged drain in the sink. And so instead of being able to then pull that glucose back out of glycogen, the glycogen just continues to accumulate and creates the Lafora bodies that drive the disease progression. And so in this second strategy, our goal is to reduce how much glucose is going into glycogen, right? And by reducing that flow, we're slowing, right, how quickly glycogen is being formed and accumulate, and that gives our bodies other ways to deal with the glycogen that is being formed. So I give you that background to help you understand some of the therapies and what we learned about their progress. So we'll start with Ion 283, which uses that second strategy. The goal is to decrease the amount of glycogen that's being made. And so what you can see here in a patient with no LD, they'll have normal glycogen storage. In a patient with Lafora disease, then you have these two enzymes that are responsible for making glycogen. And as long as they stay normally active, we're going to see Lafora bodies form. With Ion 283 treatment, it's an ASO which acts by blocking, right, the signal that tells the body to make one of these two proteins. So now the body, particularly the brain, right, is important that it's not making as much glycogen synthase. And so that slows how much glycogen is being made. And the goal of this therapy is to slow or halt glycogen accumulation so that we can slow or halt the progression of the disease. And we'll share all this information and slides with everyone after the meeting as well. So the update that we received on Ion 283, I'm going to walk through each of these updates, and as people have questions, please put them in the chat for us, and we'll make sure we get to all these questions at the end. So we received an update from our team at UT Southwest that all 10 patients have now been enrolled in the safety study. As a community, we have now raised $1.22 million out of a total $1.5 million that we need to ensure that the study is completed. And we got a bit of an update on how the therapy is going for the patients who've been treated. And right now, if you'll remember the safety study, you had to start with a very low dose of the drug, a 15 milligram dose, to test for the initial safety. So far, it has been well tolerated in patients, meaning that there haven't been any significant safety concerns. But based off of the data that they have so far, it also hasn't halted the progression of the disease. So the researchers have requested that they be allowed to administer a higher dose, a dose that is predicted to be effective at slowing the disease progression. So that's a 30 milligram dose. In order to give a higher dose of the medicine to patients, the team at UT Southwest has to receive permission from the FDA. And the FDA had asked to ensure that they had data from all 10 patients, that all 10 patients were enrolled, and that they could show both that it was not only safe in the patients, but they asked for a biomarker to be able to track disease progression before granting approval to raise the dose amount. So the team at UT Southwest, as of the symposium, was working to get all of this data together that the FDA requested, and working with us as a community to identify a biomarker that could be used for this process. Also at symposium, there was some questions about Noventia. Noventia did not give us any updates for the symposium, but the team at UT Southwest did agree to share any data that was needed or requested by the team at Noventia so that they can accelerate the clinical trial pathway in Europe as much as possible. We've not received any updates from Noventia since this summer after we submitted the open letter that many of you signed on to. You can find the open letter at our website if you want to read their updates. What we learned at Symposium is that contracts are still being negotiated. Noventia is planning to contract with Telethon in order to make the clinical trial in Italy happen, but as of the symposium, the contract had not yet been signed. So that means nothing can move forward with that trial in Italy until a contract is signed. So we're waiting for an update and news that something has been signed. I know this is a very important opportunity and an important issue to many of our families. So we do make sure that we keep our website updated. We have a special page dedicated specifically to the safety study. So always feel free to check and monitor that web page if you need updates with what we know. When we know it, we want to make sure that the community also knows as well. All right, so from the ION 283, we're now going to move to some of the therapies that are in the preclinical space and give some updates on what we learned about those drugs. So we'll talk about our antibody enzyme fusions next. So that includes the VALO-417 drug or the VAL-1221 drug. And so an antibody enzyme fusion uses an antibody, which is a small protein that's connected to a different protein. And you can think of the protein that is connected to as that pair of scissors that has the ability, right, to break apart those Lafora body aggregates. In this case, right, this other protein is called an amylase. So you may hear either amylase or a glucosidase enzyme, but their function is essentially the same. They can cut, they can chew up those Lafora bodies and release the glucose back into the cell. And so here's a picture of how this would work. The reason it's fused to an antibody, the antibody helps transport the protein that we need into the cell in order for that this green protein to do its job and degrade the Lafora bodies. So this red protein is important for transport, getting the medicine to the right place, and then the green protein is the one that's doing the work of breaking down those Lafora body aggregates. So we did get a couple of updates on what was happening with these drugs. So the VALO-417, we had previous data, previous presentations that had shown us that this drug successfully degrades Lafora bodies in the cells and can restore normal metabolism. Over this past year, the update we had was that it was given to a canine with Lafora disease using a delivery mechanism that allowed this medicine to get directly into the brain. They're still, right, collecting data, monitoring the dog, but the owner had reported that the dog appears to be in good health. We won't know more until we can really get samples from the dog, or until the dog passes in its natural death, then we could get samples, but it is someone's pet. So that does limit how much data we can collect. We do, this is right now just one dog. We do want to be able to test this in more canines to have enough data to show that this drug is effective in a large animal model. And then we would be able to use that data potentially to help us accelerate and apply for a clinical trial. A barrier that we learned about during our discussion and that we knew was potentially an issue, the IP, the intellectual property, so the rights to use and produce this drug belonged to originally Valerian. Now the company is called Paracel, which had gone bankrupt. And so the use of the drug for clinical trials would have to be negotiated, and it's very likely that the patient community would need to pay 100% of clinical costs, much like we're doing with the safety study. For Val 1221, these, again, some of this is data that we had already learned over this past year before the 2025 symposium. As I mentioned, around 8 patients have used this drug at some point through compassionate use. We knew from animal models that it was effective if you could administer it directly to the brain. Unfortunately, this drug was originally approved for use in Pompe disease. And so in Pompe disease, they're trying to treat the accumulation of glycogen in the muscles. And so they approved it for IV administration. In Lafora disease, we need the drug to get to the brain. And the data that was collected from these patients showed that while the drug did get into the bloodstream, it did not cross into the brain. So in order for it to be an effective treatment for patients with Lafora, we would need to get this drug approved for administration directly to the brain. So that would mean sending it back, right, into a few preclinical studies, trials, and then getting approval for it to be administered directly to the brain. This has the same issue as Valo 417, where the IP is owned by Valerian, now Paracel. So unless there's negotiations and a willingness for someone to move forward or for someone to sell the intellectual property rights, we can't move forward with this drug without negotiating with that particular company. There was a third antibody enzyme fusion option that we learned about at symposium this year. So this one is very similar to Valo 417, but it has a third component to it. So in Valo 417, we said there was an antibody that helped transport it into the cell. There was an enzyme, an amylase, that helped break apart the Lafora body. This particular fusion has a third part, and the third piece is this transferrin receptor, which is designed to allow it to cross the blood-brain barrier. So the implications of this, right, is that this antibody enzyme fusion drug could be administered through an IV process, and that third component would allow it to cross into the brain. Early mouse model experiments have shown that this HTFR amylase works similarly to Val 1221, Val 417, and we asked for a timeline, some estimates of how long it would take to wrap up the preclinical work. So for the other two antibody enzyme fusions, the preclinical work is essentially done. The biggest barrier is the IP issue, who owns the rights to produce the drug. With this third one, the rights for the drug are owned by University of Florida and Harvard, so we would not have the same issue with getting the drug produced, but it's still in an earlier stage. It still needs about a year of preclinical studies so that they can determine the minimum effective dose, so how much of the drug is needed, how often it needs to be administered, and also doing some toxicity studies to make sure that they know how well tolerated the drug is. What's the maximum amount that you can give? All right, so from our antibody enzyme fusion drugs, now the third kind of family of therapies that we got an update from at symposium was our gene therapies. We got an update from the Saratosa lab. They've been working to develop gene therapies to replace the EPM2A, the gene that codes for Laforin, and to replace EPM2B, the gene that codes for Malin. These are two separate therapies. They would have to be approved right through their own independent trials. So that means since about half of our community has mutations in EPM2A, they would need that first therapy. The other half has mutations in EPM2B. They would need the second therapy. So these gene therapies are designed to replace the mutated versions of the gene with a correct version. They've now trialed both of these therapy designs in mice, both of these gene therapies. They showed data at the symposium showing that EEG, the electric signals in the hippocampus, were restored to normal levels. They looked at damage to astrocytes and neurons, and they noted that the gene therapy treatment didn't show a reduction in damage to the astrocytes, so one cell type in the brain, but for the neurons, it did appear to reduce neurodegeneration in the hippocampus of the mouse, right? They were just looking in that one region. They also presented work on what they're doing to improve distribution of the drug. They have to make sure it reaches all regions of the brain, so that's an ongoing process that they're trying to optimize. Some of the barriers that we discussed, it's currently very expensive to manufacture the drug. We're talking close to $3 million just to get the drug approved to go into clinical trials. So that's before clinical trials could even start. So the expense is one barrier because it would have to be likely covered by the patient community because this particular therapy wouldn't be effective for anyone outside of our community. We also learned from a later presentation at Symposium from a study being done at UT Southwest that suggests that if you express too much Laforin, that can also be toxic. And so we would need to be careful to monitor how much expression of EPM2A is produced by a gene therapy for EPM2A for a Lafora treatment. All right, and so that takes us through those primary therapies that were that we got updates from that are in different stages of preclinical development. At this point, I would like to open the meeting if anyone has questions about the updates or any of the therapies that we presented on. You can put a question in the chat, or you can use the raise hand feature or turn on your camera and raise your hand if anyone has questions.
Kit, obviously, if there are questions, that takes priority. But since there doesn't seem to be like an urgent question, I can give some updates, and I can also kind of give some further thoughts to what you said, if
Sure.
that's okay.
Yes.
I'm going to share my screen. I prepared one slide. Can you see my screen?
Yes, we can.
So you presented the main therapeutic approaches so nicely, and they're summarized here. It's exactly what you presented, which shows that, you know, basically everyone's on the same understanding. So I've divided it into 3 sections like you did. The section 1 is how can we reduce glycogen synthase? And as you mentioned, the ion 283 is in patients. You correctly mentioned that the dose we're using now, 15 milligrams, is too low. We knew that it's going to be too low, but it was the only way to get started. We asked permission from the FDA to go to the higher dose, and I can very happily report to you and everybody that we got that permission just this last Friday. So the next patient, the FDA said any patient who received 3 times 15 milligrams can now receive 30 milligrams. So we are going to do that. So most of our patients actually have received 3 times 15 milligrams, so they will be getting 30 milligrams. Since the purpose of this meeting is to kind of focus people's attentions, on which of these studies or what studies to prioritize in supporting, I want to emphasize that it's really, really important to complete this study. The community has done an amazing job at raising like the large majority of the funds. There's a little bit left. We need to do that because this bird is in the hand, as they say, everything else is not in our hand. And who knows what problems the various other therapies will face. So we need to complete this, go to the higher dose. The nice thing with the FDA's response was they really, really understood this time with my very strong letter the situation of our families and our patients. And they opened the door to even higher and higher doses. But it has to be step by step. They want to see the effect at 30, but then they're open once they receive the data to go even higher. So at some point, we will hit the dose that we know must work because it has worked so well in the mouse models. So that's number one. Section 1.1. Now, there are other ASOs that various groups are working on. There's divalent siRNA that our lab is working on. Each of these things are advances on the classical ASO technology, but I highlighted in green what I think, and I'm obviously biased. Everyone should keep that in mind. What I think that the community should focus on. So there's just not enough resources to support everything, obviously. So for the reducing glycogen synthase section, I think we should continue supporting the ION 283 project to completion. Yeah. Now, the second section, like you presented, I put here, is getting rid of Lafora bodies, digesting the Lafora bodies. And this is all based around the VAL-0417 kind of technology. As you mentioned, we treated one dog, and the owner of that dog said the dog is doing well. She said it's doing better. But one cannot draw scientific conclusions, from one animal and, via one owner's report. So we were nonetheless very excited with this report. At least we know that the drug delivered directly into the brain in the CSF. What's done here is it's put in the CSF space in the dog just behind the brain. It's very easy to do that in dogs, unlike in humans. The veterinarian holds the dog up by the nape and puts the needle in. The space is so large that they can put the infusion. So it's a very easy experiment. So now with our German colleagues in Leipzig, we have 4 more dogs lined up. And we really want to do these next dogs. And we're going to be analyzing them with extreme detail, including myoclonus measurements, maybe EEG, but motor examination, all sorts of things, kind of like a clinical trial. And we're going to double the dose. So we're hopeful that with the 4 more dogs and with very close evaluation, we can reach a conclusion whether this actually gets rid of the symptoms in the dog. The dog, unlike the mouse, is more like the human. It has all the seizures and everything that Lafora patients have. So we should be able to know if by getting rid of the Lafora bodies, we got rid of the symptoms. So you mentioned that there's intellectual property concerns with this. That is true, but those concerns are only in the United States. This company that owns the intellectual property of this has not protected these rights in Europe. So we're free to proceed to develop a drug in Europe. And that's a big advantage given, anyway, most of the patients are in the old continent in Europe and places like that. So for this to proceed, it requires funding. I'm not today asking for money, but I'm just saying where this stands is it requires funding. And it's kind of on hold right now until we can raise the funding. The biggest cost is producing the drug. The cost of the experiments with the dogs is very low. Our German colleagues and the neurologists there and so on are ready to do most of it for a very, very small price. But the cost is in producing the drug. I'm happy to share the exact cost later if need be. So I think the community should support this because it's a very quick answer for whether VAL-0417 put directly in the brain will work. As you mentioned, there are next generation technologies that Dr. Gentry is developing. Also, there's a variant that Dr. Nitschke is developing. But those, they all deserve to be supported, but they're at kind of earlier stages. And for a community that is focused on getting quicker treatments, the priority, I think, in this category would be to complete that project in the dogs. Finally, gene therapy. The beauty of gene therapy is that we know our patients are missing a gene. We put the gene back, we make the brain whole again. And in principle, it's the most simple to understand and straightforward approach. And obviously, I support gene therapy in a very big way. I have put 2 points. The number 1 point is the way that we've all done gene therapy until now with the existing classical viruses. It's very well known in gene therapy, which way you try to put it in the brain, the current viruses, you only transduce, meaning you only correct about 10 to 20% of cells. So the results of gene therapy can be very nice, but they're always very, very partial. In Lafora disease, we need to treat the entire brain. So it needs a leap forward towards new technology. That new technology is here now, and that is the section under 3.2. A new virus was invented, let's say, in Boston, in one of the Harvard schools. It's called CAPX-AAV9. Just like you described with the transferrin receptor that Dr. Gentry is working on to cross the VAL products into the brain, that same tag is placed in the virus. And you put the virus in the blood, and it goes into the brain. The results with this are absolutely stunning because almost the entirety of the brain gets corrected. Why? Because the blood system reaches every single cell in the brain. And so the virus can reach every single cell through the blood system. And there's basically no blood-brain barrier for this virus using this technology, and every cell is corrected. This is the modern day way of doing gene therapy. The whole field of gene therapy is now shifting from our standard viruses to this virus. So I think the community should support us developing this. And by us, I don't mean my lab, although we are doing this, we're happy to do it. I think maybe driven by the community, we can all come together and do this together, all the groups that are interested in doing gene therapy. There may be a way to do this all together. So I would support using the best and newest virus rather than the older viruses. One final point, this virus crosses the blood-brain barrier so efficiently that the doses you have to place in the blood are much, much lower than what we have to do with the old virus. And the prices for this drop from the millions to the 100,000s and very quickly to the 10,000s. It's not exactly there yet, but it is expected that in the next year or soon after that, the cost of producing these viruses for human studies is not going to be in the millions, probably in the 100,000s. There's even estimations that it could be in the tens of thousands. So the huge hurdle of virus production costs is going to be very different with the modern viruses. I will stop here, and with your guidance, happy to take questions or do whatever you want to do.
Yeah, Dr. Menashe, there were a couple of clarification points. One question was going back to Ion 283 about the dosage, and you already kind of mentioned that this is a kind of a stepwise process, stepping up based off of the data that was collected when you were testing this in the animal models. Do we expect that 30 milligrams has the power to start showing some slowing, or is it too early to tell if 30 milligrams is gonna?
Yeah, it's too early to know. It's certainly double the 15 milligrams and in the correct direction. And you know, we're trying to slow the natural progression of the disease. Even if we slow it a little bit, that's something. We know that there's a certain dose that will stop the disease, but whether we can get there, you know, depends on how safe each of these escalation steps are. So I am hopeful for the 30 milligrams, but I cannot know for sure.
Okay. The other question was about Novention. I don't know if any of our other patient organizations or if any of our Italian clinicians have any updates on whether or not any contract was signed with Noventia. But initially, the letter in the summer had mentioned early 2026. But if none of the paperwork's been submitted to the EMA, our understanding is that's about a year-long process, can be, to get everything approved. So so if nothing's been submitted yet, I'd say we're looking at least a year out from expanded trials in Italy. Giuseppe, did you have anything to add?
No, not really. I don't have anything to add. I talked very recently to somebody in telephone, and the contract has still been not signed. There's some sort of I don't know. They can reach an agreement, and we are all waiting, and nothing new. Actually, nothing new from what we said in Valencia. There's nothing new.
Okay, thank you. Okay, and a clarification, a question was, and this goes back to you, Dr. Menashe, is there any hope of expanding the study in the US to give more people access to Ion 283?
Um, it's a complicated question, and again, I preface it by saying that I have like zero power because this drug belongs to Ionis and they licensed it to Noventia. So it's between Ionis and Noventia. I can tell you I have a little update in that we are having a meeting, including myself, Noventia, Teleton, and Ionis coming up. I think it's next week sometime. You know, mostly to the agenda is to kind of, I guess, discuss the development of the study in Italy and sharing our data with them and so on. So the fact that this meeting is set up gives me hope that people are engaged and something is happening, but I don't know anything beyond that.
And we've got another question about biomarkers. So anyone involved in the biomarker space can feel free to chime in. So since the FDA is requiring, and any regulatory agency is going to require a biomarker for any of the therapies for us to be able to prove that it's effective, So do we have any way to measure, it says glycogen synthesis right now, I'm taking that to mean changes in glycogen accumulation. Do we have a way to track that right now in patients?
We don't have something completed and clear, but at the meeting, as you know, Kit and everybody, there are many excellent biomarkers in the works. So though I know that Kit is organizing, bringing all this together. So to answer in general terms, I think we have very promising biomarkers, which will probably become actual useful biomarkers soon. I think maybe in the 6 months or 1 year. Now, I want people to understand something. Biomarker tracks the disease progression. So if we can, for example, measure the amount of glycogen in the brain, either in the CSF or by MRI machines or some other way, it will help tell us that, okay, more glycogen is not being made. The glycogen amounts have been stabilized. So that's a biomarker of the disease. What we need urgently, more urgently than the biomarker, is something called target engagement assay. So in the case of the ion 283, our target is the enzyme glycogen synthase. And you asked the question, Kit, will the 30 milligrams work? The reason I cannot answer is because we don't have a target engagement assay, meaning we have to give the drug and wait and see what happens to the patient. But if we could measure the target, which is glycogen synthase, in the CSF, then we could know by how much we decreased the target, the glycogen synthase. Then we can extrapolate to our mouse results, and then we can predict what's going to happen. So we're very anxious to develop the target engagement assay. This was initiated with Ionis in the past, but then stopped when they stopped working on this. We have restarted this and are working with a group in Germany, in Munich. They have very fine technologies where the enzyme glycogen synthase can be measured with two antibodies, sandwich ELISA, to which a piece of DNA is attached, and you do PCR on it. For those who can follow, it means very, very low amounts of glycogen synthase can be quantified. And so we are doing that work, and in fact are using the samples from the current study, because every time a patient comes, they give us CSF so that we can get biomarkers. We're using that CSF to see if we can finalize this target engagement assay. This is so important that the FDA itself, in their letter to me last week, said, please focus on completing a target engagement assay to help us help you determine what level and what dose you can give. So this is a very important thing that we're working on actively right now.
Yeah. Thank you for those updates, Dr. Menashin. We only have a few minutes left. We do want to continue to respond to any questions from the community about where different therapies are at in development. So if you don't get to ask a question today, we do have a survey that we're asking families to complete based off of what you learned today. Help us, and I believe, yes, you can access the form directly through the link in the chat right now. We'll also send out a follow-up email with the recording from today, along with the link to this form. We really do want to hear from the whole community if you have additional questions, but it also asks for your feedback based off of what you've learned. Which therapies do you think are most promising for our community? Where would you like to see us focus our efforts? Dr. Menashin highlighted, right, his thoughts on the therapies in the different spaces, the different modalities. We're a small community. Supporting all of them at the same level is just not going to be possible. We want your input to help us determine what to give the strongest support to. And it doesn't mean we ignore the others, but we do need to prioritize. If we think that there's a therapy, if we hear from you all that there is a therapy that we really want to push into the clinic and help support to ensure that we can get more of our therapies into the clinical space to treat our children with Lafora. All right. So if there are no other questions, I will remind people, please fill out the form. We do need to hear feedback from you. We are planning to have another meeting based off of your feedback that will be more of a strategy session where we can talk practicality based off of what we hear from you. We'll research what are some numbers, what's the cost with supporting certain therapies. We'll get the numbers from Dr. Menashin, what it would cost to finish out that canine study for Valo 417. That way we can meet as a community and decide what ways that we can financially support, what ways that we can offer other kinds of support, or if we need to assist with collection of materials or data that would be useful for supporting any of these projects to move them forward through the pipeline. So thank you all for being here. And be on the lookout for the email from us. I will also share a PDF version of the slides that I presented so you can go back in and see the updates that were presented as well. And as always, please reach out if you have any questions. Thank you all so much.
Thank you. Bye.